Well, good day, everybody. My name's Martin Kiernan, and it's my great pleasure to host today's teleclass. Today's speaker is Professor Keith Warriner, and he's from the Department of Food Science at the University of Guelph. Professor Warriner's journey into food science began after his initial training, but he worked originally as a chef in the UK. He then got his first degree in food science from the University of Nottingham, PhD in microbial physiology from the University College Wales, did some postdoctoral work on biosensors at the University of Manchester, and then another research fellowship in food microbiology at the University of Nottingham. So he certainly toured the UK for a period. But in 2002, he joined the University of Guelph, where he was promoted to full professor in 2011. Throughout his very long and distinguished career, Professor Warriner has made significant contributions to the field of food safety and microbiology, and his research focuses on enhancing food safety within meat processing and the fresh cut sectors. Also has been involved in the development of biosensor devices for detecting foodborne hazards. He's published over 100 papers, book chapters, patents, and conference abstracts. And this reflects his huge expertise and commitment to advancing food safety. And I know listeners in Canada, if there's ever any national food incidents, then he's bound to be the person that the media turn to for comment. So today, Keith's going to share his expertise on food safety of fresh produce, which he said is an old food safety problem, but with new solutions. And I do like to hear that there's a solution to a problem. So please join me in welcoming Keith. Thank you very much, Keith.
Hello, thank you for that, Martin. It's good of you to introduce me. So, yes, I've certainly had a long career in food microbiology, and not ironically, but coincidentally, I started my food microbiology research with fresh produce. And, going back 30 years, I suppose when you look to the future, although you don't kind of look to the future when you're so young, I suppose, but when you do look to the future, you think things are going to be changed, things are going to be different, and we're going to move on to another food safety issue. But the reality is with fresh produce, you almost can say where we were 30 years ago is where we are now. There's really been no real improvements. Obviously, the detection, the prevalence of pathogens in growing areas have increased, but the way we sort of act towards increasing food safety hasn't really changed. And then what I'll do is I'm going to talk about the history of fresh produce safety, not go through outbreak after outbreak, because it does get a bit weary after a time, but really to illustrate why I'm saying it hasn't changed much. I'm going to talk about the spate of romaine lettuce outbreaks we had here in North America, it's actually went back from 2016 up to even last year, I believe. But we'll talk about how some of those outbreaks just went under the radar. And a recent one, just early last year, the FDA totally covered up, and I'll tell you why that is as we go along. I'll talk a little about the pathogens of concern. I think they're known to everyone, but I'll just say how they've broadened and the number of targets have increased. And then talk about this risk mitigation, which has been very slowly adopted within the industry. And we'll talk about why that is. And then I'll talk a bit about our own research on how we've tried to improve things. Because we started research on intervention methods way back in about 1999. And it's come to fruition in commercial practice just recently. So for those old enough, and I don't think there's many old enough these days, to go back, back in the 90s or the 80s and the 70s, fresh produce. So if you got lettuce and things like that, you knew it was summertime because you don't get lettuce in the winter. It was fairly all local, very narrow selection, a few baskets, if that. And back then, they had so many food safety risks with everything else going on. Obviously, the listeria in poultry, or the salmonella in poultry, salmonella in eggs, and things like that. Fresh produce was fairly low on the ground in terms of food safety risk. But the reality is, I can remember the first outbreak with lettuce being reported back in 1970. Not remember as in I was there because I was only young myself. But when you look through the records, that was, I think, the first one. And then you used to get sporadic ones, or we had a lettuce outbreak at McDonald's or things like that. But it wasn't really until the big sprout-related outbreak in Japan and that affected thousands of people with E. coli 157. It was to do with radish sprouts at that time, and it really brought the industry up. The sprouted seed people got kind of, what's the word, separated out from the fresh produce people, saying the sprouted seeds are so dangerous, and people have that perception now. But they always thought the other produce was safer, but obviously it didn't turn like that. But there was a really big revolution in the 90s as well, apart from the big outbreak in Japan, and that's the start of ready-to-eat salads. So back in 1994, Marks and Spencer's came out with the first bag salad. Obviously, people thought it was very convenient, and Marks and Spencer's always were the front runners. And it was convenient to use, extra shelf life, debatable, but people say that. But the increase in produce availability also all year round supply, getting it from different locations. And back then it was starting to have these centralized production. So in North America, about 80% of the lettuce we get is from California, including Arizona. And they have this sort of seasonal growth cycle. So obviously, in the summer months, it's too hot in Arizona and Southern California. So it really moves up to Central California, Salinas Valley, which is important to know for the next few slides. And then as the fall comes, it goes down to central or the southern part of California and Arizona. But basically, vast fields. If you ever went to Salinas Valley, you'll just see vast fields of lettuce and things like that. And they're global sourcing, so we get a lot of produce from Mexico and things like this, and other countries which you assume they're taking the same food safety precautions as you are, but it's not always the case, as we'll see. So the reality is, in North America at least, not so much in Europe, even though I think in Europe it's definitely is, just like they don't report it, is that fresh produce is the number one cause of foodborne illness outbreaks. So it's not the cause of number of illnesses, that's still resides with meat, but it's the outbreaks. And this really is a result of a heavy centralization. So we don't get small outbreaks with fresh produce these days. It's usually multi-state, multi-province, and a lot of people involved, and sometimes deaths. So the reason why it's so susceptible is that we consume it raw. Nobody heats a lemon, a lettuce, and things like that. And it's an open field, and that's open to a lot of contamination sources. And down in California, and Yuma, and we can include those in it, they have feedlots and animal production close by. In the Salinas Valley, for example, it literally is a valley. So you've got on the valley floor, you've got the fresh produce growing, and you've got ranches on the hills. And you know, when every time we talk about ranches to fresh produce people, they said they should move at least 10 miles back. And obviously, the ranch people don't quite appreciate that since it's been there longer. And they say so. I was in a meeting a few years ago now where they tried to get ranchers and fresh produce people together, but let's say weren't productive. They didn't really talk to each other that much. More accusations. So the thing is, as Martin said, when there's a fresh produce outbreak or any sort of outbreak, I'm usually contacted by the press or the media. And the thing, good, well, I'll say it's a good thing in the Canada. They don't try to be so secretive about it. You know, they know that consumers must be aware. And I remember back in the day in the UK, it was very seldom we would talk about food safety issues and things like this. But in Canada, in North America in general, it is certainly on the news when there's an outbreak. Well, depending on how it's impactful. But obviously, this erodes consumer confidence. And just last year alone, we had outbreaks of cantaloupe again, to do with salmonella, listeria outbreaks. We've had cryptosporidium, norovirus outbreaks, and things like this. And this really reflects how the industry goes, is that they seem to have outbreaks and not be able to control them. Now, the thing is just recently even, there was an outbreak of salmonella with cucumbers. This was down in the US, and they found it was a same contamination source. We're getting increases of outbreaks associated with Shigella, Yersinia, even the bag salads, which again, you know, people perceive as safe because they're washed. The reality is industrial washing doesn't do anything. So the reality is every year we seem to be getting repeated history. Nothing seems to change. And even in the UK now, they're starting to detect more outbreaks linked to lettuce. And that's unusual because in the UK, when they do have outbreaks, they tend to be what's the word less detailed than do in North America. So a lot of outbreak goes unnoticed. But again, because of the surveillance systems and detection systems, more likely to be detected. Now, as I mentioned to you, the sources of contamination have been known for a very long time. Uh, the main source of contamination is typically from irrigation water. And, we talked about how animal production seems to be close by crop production. It's just the way of the nature. So we get manure runoff into irrigation canals, and those irrigation canals are like veins. They serve a wide area. And as you can imagine from the climate, they need a lot of irrigation, apart when it's flooding. And that's the other part of it. We're getting much more extreme weather events down in the growing region. So we get flood events, which spreads contamination, but we can get contaminated irrigation water. And with the feedlot farms, they obviously produce a lot of manure, and it runs off, and the insects come and transfer contamination. We've had animal intrusion. But once the pathogens like E. coli and Salmonella come associated with plants, uh, they're adapted to colonize it essentially and survive. And it came as a surprise back in the 90s when we started researching this in terms of we've always thought plants were the sort of hostile host to a E. coli, which is used to living in the GI tract. But what we showed, I won't go through it too detailed, is that yes, you can get internalization. Yes, E. coli does colonize the roots and it can contaminate seed. We talked about the sprouted seed people. 98% of the outbreaks associated with sprouts, like bean sprouts, alfalfa, are to do with contaminated seed. So we get all these sorts of contamination, which obviously have to be controlled. So the problem is, though, is that the fresh produce market really didn't take food safety that seriously. They were more interested in growth, and they certainly did grow, and it still grows today, 10% a year, especially the bags salad market. Uh, Obviously, a greater diversity of produce. They're getting it from different countries, and it's all bigger and better. But the thing with the fresh produce industry, they're always low margins. It's not a very rich industry. That's why you need the volume. And I'm not sure what the tariffs are going to do, but that's another matter for another day. So, to illustrate how the industry really look at fresh produce problems in food safety, I think we just need to go back to the spate of outbreaks. So, officially, there were 8 outbreaks over 4 years to do with lettuce from California and 1 from Arizona. But in reality, it goes back longer than that. So, back in November 2016, there was this sort of mystery outbreak of E. coli 157. They never did trace the source. All they could do is get the fingerprint and collect cases together. But the same isolate turned up in 2017. And this was to do with romaine lettuce, and romaine lettuce produced in uh, California, well, the central California, Salinas Valley to be specific. But it was a mystery to them at the time where it was coming from. And the reason for that is because they were very difficult in tracing back because lettuce has a short shelf life. So, and people don't remember what lettuce they ate and things like that. But then the second outbreak happened, and this was from Arizona. And the only reason they could trace it back to Arizona is because an outbreak occurred in an Alaskan prison, of all places. And in Alaska, obviously, you've got a contained environment, so you know exactly where the lettuce came from. So they actually could say, oh, yes, this lettuce is from Arizona. Now, the thing is, they tried to do trace back, and they were getting linking cases with the DNA fingerprints of the E. coli. But the reality is that it was a very confusing picture. And this is this sort of map the FDA drew up. Obviously, you're working backwards, so you look to see where the user level is, but then you've got to go for the sort of distributor, then the processor, and the the farm and the harvester, because one of the big things in the fresh produce industry in the US, well, in Canada as well, it's all subcontracting. So they subcontract growers, who subcontract harvesters, who will subcontract processors and probably distributors. And you can see the flexibility in this. So they can literally source lettuce all the time when needed. But when it comes to an outbreak, obviously traceability goes all out the window, because you don't, it's all commingled. You're not really sure where the produce was coming from or where's it going to. Now, so they wanted to do is try to isolate a farm, and they couldn't, because there were so many different farms involved. They said, well, it must be a common source then. And someone, I don't know who it was, said, let's sample the irrigation ponds, the sediments especially, where the E. coli resides. And sure enough, they started getting hits of E. coli 157, and this is in Arizona, the Yuma Valley. And essentially, we're getting hits, and it was matching up. And they said, yes, that's it. There was a common irrigation water source. And they did further trace back to see where the E. coli could potentially come from. And as you probably guessed by now, it was the feedlots. The feedlots were producing manure, which was then running off into the irrigation canals, which were going through the Yuma Valley to do, and people taking water from them. You're allowed certain water rights and that, so you've got no choice. So you've got to use it because there's no other source. And they irrigate the crops. And where romaine lettuce, people say it's more susceptible, because essentially the tissue's more delicate and it's not loaded with antimicrobials like anthocyanins. Back then, they noticed kale was, for example, irrigated by the same water, but they didn't have an outbreak with kale. I suspect it's more because they don't you don't eat kale raw, but they suggest it's to do with antimicrobial of anthocyanins. So the thing was, you could imagine back in 2018, they had two major outbreaks, multi-state outbreaks, and the industry had to try and get confident. And I think any other industry would have said, like, let's clean up the ag, let's clean up the irrigation water, let's get interventions where we can pasteurize the product essentially before distributing. But the fish produce industry don't work like that. What they did is they got the Dr. Oz, still going, I think, and they had this sort of fresh fat salad summit, and they had all the sort of leaders there. I know Dory from Dole, I think. Oh, it's mentioned names, but the reality is they got these major processors to try and calm consumer confidence. And they came on saying how safe salad was and how the industry is striving and learning from mistakes, and people are totally safe to eat salad. Well, a few months later, they were kind of proven wrong, for the simple reason we had another outbreak. So this is the third outbreak now, what we know about. And this was based in the Salinas Valley. And as you could imagine, at this time, there was a lot of panic. Not so much from the public, oh, I suppose it was from the public, but from retailers. They say we're going to remove every romaine lettuce in the market. And as I mentioned to you, they were confident that they could do this because most of the lettuce at that time would have come from the Salinas Valley. So they removed everything in Canada, even. We couldn't get a romaine lettuce from anywhere. And so it was a persistent problem. But more worryingly, what they found is that strain that was from 2016, and then 2017 was also in the 2018. And they managed to isolate the E. coli. And it was just characterized a few years ago, actually. It's not very long ago. And they found it had been adapted to the environment. An E. coli 157 that could grow in the environment, which was unheard of before. You know, the big assumption was E. coli's in manure survival mode, doesn't grow. But this one was. And even more worryingly, it was found in the aquifer in the Salinas Valley, which basically serves about 300 miles of cropland. But anyway, so this E. coli obviously evolved to persist. And so basically, after the sort of fourth outbreak, it was even more pressure on them. This wasn't going to be a Dr. Oz kind of conference on TV to calm people's nerves. And really, what was happening is that the industry was kind of trying to suggest that they were learning from it. The FDA were getting a bit tired, or the LGMA doing, and I'll talk about the LGMA in a little bit, say we have to do something. We have to try and improve food safety. But the FDA's attitude to food safety of fresh produce and most other things is the industry is responsible. The industry is responsible for controlling food safety. We're just here to maybe investigate it, but and maybe give a few guidelines. We're not here to make sure it's safe. And so the FDA kind of started washing the hands of the fresh produce area. But they did an investigation with the CDC, and they actually managed to trace it back to a farm in Santa Maria, and which is in the Salinas Valley. And this was the Adam brothers. And when they actually looked at their operations, basically they do use irrigation water. They actually had a feedlot in with the farm, and they weren't doing any sort of test. Well, we're doing testing, but not reacting on it. So they were getting fecal coliforms, E. coli in the water, but not acting on it. And you might think, well, why did you do testing? But this is again their philosophy is that they test to expecting to find a negative, and when they find a positive, they're not really sure what to do, and that's very common even today. And they were trying to use chlorine, but it wasn't working. I think it was too dilute and things like that. So it was a bit of a disaster, but at least they found the source of it. Now, the thing was at that time is that the fresh produce industry were in defensive mode still. You know, you could imagine after 4 outbreaks. And Jim, God rest his soul, he died 2 years ago. Um, he was a very good he was a spokesperson for the fresh produce industry. And he was trying to convince people that even though we have outbreaks, we have so much lettuce, like 1 or 2 outbreaks doesn't really matter. It was a total disaster because if anyone knows, even today, if you said, well, there's only 20 people who's been affected and 3 died, you wouldn't get much thing. So, but this were very defensive modes. And really, what was their big defense was what the LGMA were doing. So, a brief history of the LGMA, Leafy Greens Marketing Agreement. Back in 2006, there was an outbreak of E. coli 157 associated with spinach, and it was 200 people. I think 5 died, and it decimated the spinach industry. That took, I think, it was 7 years to get back up. And they developed the LGMA back in 2006 in order to improve people's confidence, and they would come up with guidelines. And these guidelines would be test your water once a season, have a look to see if there's animals there. It was kind of a flimsy standards, which the membership, should we say, were very okay with, because it doesn't mean much testing. It doesn't really mean much, but to the outside, it looks like the industry is taking care of itself. So the LGMA at the end is the big one, the marketing agreement. They basically said if you don't follow our standards, you don't supply the market. And like I say, these standards were very loose indeed. And when these outbreaks occurred, you know, this was over 10 years after the spinach outbreak, all they could come up with is saying, well, thank you for the FDA for identifying the problem, the source of the root. Yeah, we're going to do all we can to take this learning process and move forward to make sure that produce is safe later on. Now, the thing is that I think it was last year, I always forget, there was a documentary which a lot of people might not have seen. I don't know if it was launched anywhere other than the US. There's a program called Poisoned. So Poisoned was this documentary. I actually went down to Hollywood. It was outside Hollywood, be outside Hollywood, to do an interview. And this documentary team were trying to obviously look at the state of food safety in North America. And had me there for 4 hours, and they were kind of quizzing me, saying, well, what's wrong with the fresh produce industry? What needs to be done? And, you know, I thought I gave some good points, as I'm doing, I guess, as we do, you know, saying how it's a very weak industry, how it's not really focused on food safety. Then I got an email a few months later saying, oh, we're not going to use your bit. And I thought, that's a bit ungrateful. I know it's not so good, but it's not so bad. But when I saw the documentary, I saw why they didn't use it. So the reason why they didn't use it is that they had Tim York from the LGMA. He was the head man, and they asked him a few simple questions. And some of the questions were, has the LGMA standards actually improved food safety? How many samples of irrigation water did it take? Yeah, I kind of fed these questions to them in a way. And if you saw the program, basically Tim York had no answer. He can't, and I did warn him about this actually, that he shouldn't have done it, but he did it, and it came off worse. And I must admit, I've seen Tim since. I think he's still the head of the LGMA, but it's not. His team's a low profile. But the point was, the LGMA was exposed for not being able to have effective food safety systems. And yeah, the reality is, would it have got worse if it wasn't for the LGMA? To be honest, I don't think it did. But the problem was is the FDA were really relying on the LGMA to ensure food safety. But then these outbreaks started to continue. And one of the most striking sort of cases was a poor young boy who was in Disneyland in California. And he suddenly got sick. You know, he basically started getting sick. And if you've been to the US, you'll know about medical bills. And you could say, well, they should have sought medical attention right away. But medical bills in the US, especially if you're a visitor, isn't good because they're from Canada. So anyway, they tried to make it up to Canada, Vancouver again. And in Seattle, the poor boy got even worse. So they did go to a doctor and he said, oh, make sure you take him to the hospital. So long story short is that he had advanced E. coli 157 and he went into hospital. And basically, when you're in hospital, they can't really do much for you. If they give you antibiotics, that stimulates the toxin production because it's a prophage. And he got paralyzed by it, and they got a big payout. But you know, the reality is, I think he would like his life back. But these are really shocking stories that really brought it home. So with these stories going on, you know, the industry, rather than just giving sound bites, had this sort of need to say, yes, I think we do need new technology. And the reality is some companies do. They embrace new technologies. Others, and I could give you a few examples, but I think I'll be getting into trouble, stick to the old dogma. They think they're doing all right. They'll keep going on until there's an outbreak. And the thing about North America is that when you do have outbreaks, there's a litigation lawyer called Bill Marler who basically turns up. And it's a debate saying, you know, does this sort of outbreak claims, you know, these sort of cases that they bring up, hundreds of millions of dollars payout help food safety because either you live through it or you basically go bankrupt. And it's a debate that does litigation enhance food safety. I go either way. I think sometimes you have to get rid of a bad place, but the point is that you have to go beyond these sound bites. You have to go beyond. We're going to test more because testing doesn't do anything. So there's options available to do with fresh produce. Obviously, one of them is regulation. Introduce regulation so the industry have to abide by them. Now, in North America, they love regulation. They've got more regulation than anyone else, and that's why they kind of say, oh, they must have the safest food safety system. No, we've got the most regulated food safety system. That doesn't even say that actually enforced. Now, the thing with regulations, it does impose a burden on industry and a burden on the government because the government need to enforce these. But we'll talk about them later on. The latest trend is let's go indoors. We know the outside is totally full of contamination sources. So let's go indoor farming. We're protected. Well, we've had about 2 or 3 outbreaks of Listeria salmonella linked to indoor farm crops. And unfortunately, those companies involved in indoor farm crops that were implicated in these outbreaks went bust because they couldn't get the sort of legal cases just went them. But we'll talk about indoor farming. And what we're into really is in alternative interventions, alternative to washing that doesn't do anything. So regulations, as you know, government don't like to impose them because every time you have a regulation, you have to have an inspection, you have to train inspectors, you have to tell the industry tell you it's a burden. But I think what it does in North America, especially, is give you a legal framework to do something, recall, close down. It also inadvertently establishes boundaries for business. And when I say boundaries, industry goes to boundaries. And I'll talk a little bit about companies where they're the biggest, the multinational, and they hang on these regulations. Like a F1 team would look at regulations for race cars or rules in football. They look at these saying, "There's the boundary." And the trouble when you set boundaries, people go up to the boundary, but they won't go over that boundary in terms of enhancing safety. But obviously, governments need to show that they're protecting consumers and obviously protecting the environment now. Everyone's getting on the environment, but it is a rule book. And again, it's one of these debates. Does the number of inspectors equal food safety? And it's not always the case. Now, the thing is that to have any regulation, you must base it on global standards, because if you have one rule for one country and another for another country, that's considered a trade barrier. And the thing about regulations, we can trace them all back to Codex Alimentarius, which is a branch of the United Nations. So, long story short, after the war, World War II, that is, not the other wars, United Nations were established, and they said this is a good opportunity to get these training standards in. And so what they did is got the FAO, which is more to food security. They've got the World Health Organization, which is obviously public health, the World Trade Organization form Codex, and these form the regulations, the basically standard global global regulation, but standards to make regulations in different countries. And the point is that when we look at FSMA, the Food Safety Modernization Act, it's not something just the US suddenly thought up. It's actually based on Codex standards, and people interpret Codex standards differently. Obviously, in Europe, much less regulatory sort of involvement, but in the US, alike regulations. So, because fresh produce was the number one cause of foodborne illness outbreaks, the first rule in FSMA, which was basically brought into law in 2015, was the fresh produce rule. And this was all to do with managing biological and organic amendments, i.e. manure, excluding wildlife, because obviously the spinach outbreak back in 2006 was supposed to be caused by wild pigs. They do a lot of worker health. They always blame workers, saying, "Oh, those workers don't wash their hands, they don't wash nets or hair nets or things like that." Obviously, integrity of buildings, and the big one was the agricultural rule. And as I mentioned to you, the agricultural rules all to do with the quality of water. The thing is, water is the main source of pathogens, as I mentioned. The sprout growers were again kept separate, saying, "Well, they're the danger ones, so they've got their own separate rules." But really, what these FSMA was doing is that they were putting the onus on the grower to do their own risk analysis. And they said, do your own risk analysis, but you could be exempt if you include a validation kill step. Well, I think they were still thinking about the risk analysis part, because as the people, I guess, in the audience today, risk analysis isn't easy. It's a lot of judgment calls, a lot of knowledge vacuums where you have to take your best guess. And the FDA were just leaving them hanging, essentially. And the LGMA were trying to do something, but they were kind of protecting their members at the same time. But anyway, so this risk assessment that you need to do location, i.e. close to a feedlot, the water distribution centre, are you on surface water, you've got wells, are those wells contaminated? The nearby sources, manure heaps and animal activity, urban centres even. Overhead or trickle irrigation, obviously overhead irrigation is more dangerous, it directs the crop. And they had this sort of idea, which I always thought was a bit crazy, is that the withdrawal time of irrigation water. So the word is that if you don't overhead irrigate your crop in the last 7 days, you should be good. But you can certainly irrigate it still, because you need to, or your lettuce will definitely dry out. But it was just saying that if you trickle irrigate, you're fine. But if you overhead irrigate, you're not. It doesn't make sense. It does make certain sense, but I wouldn't want E. coli 5-7 anywhere near the crop. But very recently, we've had lots of storms. The J came off a drought. Obviously, the wildfires in California as well is going to, all these kind of things. Is the crop going to be susceptible? But the interesting part was with a post pre-harvest agricultural rule, they removed these irrigation water standards. So before, they used to use recreational water standards, and they weren't useful, because for recreation, what they should have done is potable water standards, but they said, oh no, it's too hard to reach that. So they just said, rather than do an alternative, they just say, oh, take the testing out of the equation, you'll be all right. Again, it don't make any sense to me, but it's sense to them. So what they do though is they gave this sort of option of validated mitigation methods, i.e., water disinfection methods or produce decontamination methods. And despite the long history, the fact that we've had drinking water for over 100 years, they said, well, how do we disinfect water? Now, even I know that I'm not an engineer. I know chlorination, the lack of peracetic acid for some reason. I suppose with chlorine, it's a disinfection byproduct, but peracetic acid is favoured because it's organic certified. As a sanitizer, I don't think it's the best one. Obviously, the best technique is UV light, but the problem is that when they were trying to validate UV light, they didn't take into account that if you got cloudy water, you can't use UV because it's too turbid. And rather than getting solutions to reduce turbidity, they just said, let's forget about UV water. And they also, let's forget about Listeria monocytogenes in risk assessment. They'll rather dodge things rather than address things. Anyhow, so to move the story on a bit, they came out with this sort of well, I was going to say helpful diagram, but I don't say that. I'd be optimistic. If I got this, if I knew nothing about risk assessment or risk analysis, I'm not sure I would do it. But essentially, what it's saying, if you can't read the print, is saying, right, so you've got to look at the water source. You've got to tell us if you've got sources of contamination, what sort of crops, what sort of irrigation. And you go down this decision tree. So the first decision saying, is your water actually safe? If you say, yes or no. Is again? It's this wording, isn't it? Is water not safe or not adequately? So basically, it's like a reverse question. Basically, if it isn't adequate, you say, "Well, don't use it." That's the advice to give. But then they talk about if it is safe, is there any sort of adjacent source of contamination? A feedlot farm, and you say yes. Oh well, if you do, you need to disinfect your irrigation water. And if you say, no, I can't see any feedlot farms, say, well, could it potentially be contaminated? I.e., is there a farm maybe 15 miles up the river that could contaminate the irrigation canal? And if you say yes, they say, well, you can do testing, which doesn't work, or you can have a mitigation method. But the important thing is this is all to do with the farmer's risk assessment. The FDA give you no help apart from that, and it can get very difficult. And if you make regulations difficult, they're not going to be implemented or agreed upon. So that's regulation. Like I say, people have its critics, but other people say no, it's the best thing that could happen. But as I mentioned, with increasing areas is the indoor farming or controlled environmental agriculture or vertical farming. So just as a plug, we actually just finished a sort of white paper on food safety guidelines in indoor farming with the FAO. It's actually due out next month, hopefully, after a few sort of illustrations need to be done. But what this does is really show the complexity of the indoor farming sector and also risk mitigation methods. Because I think with indoor farming, you've got this sort of head start. You can say that with the fresh produce, they've been growing crops for tens or maybe even over 100 years, and they were very hard to change. But with indoor farming, they're kind of really just becoming established because even though we've always had greenhouses, um, they haven't been commercially viable. So, for example, in Canada, we import 80% of the lettuce, if not more. Oh, well, fresh produce, not just lettuce, fresh produce. And but the thing with indoor farming, it's had a long history, way back to Emperor Tiberius, who was recommended to eat a cucumber a day for his health. And so they had to find a way to grow cucumbers all year round. And so the first indoor farmer used to wheel out the cucumbers in the day, take them in at night. Then obviously things progress, and the Velino greenhouses, which I think were developed in the 1930s, 1950s, they really established growing crops indoors as a mainstream business. And if you go to Spain, where the big growing areas are, you'll just see these greenhouses one after another. We have the same thing in Windsor down over here. But the vision of indoor farming, I think, was always to do, can we grow crops at the most inhospitable places? Like up north, for example, where a lettuce would literally cost you $15, which is around £10, things like that. And the reason why it became commercially viable, we even got a few facilities here in Guelph, one of the biggest ones actually in Canada, is because of the technology advancements. LED lights to give light, certain engineering aspects in terms of heating and cooling, and water recycling technologies. So now it's become commercially viable. So the benefits, the vision of why people put so many millions of dollars into indoor farming is to increase productivity. So rather than be able to grow on a season, now you could grow all year round, stack the plants up. You're going to reduce food insecurity, and I suppose in the current climate, that's quite good. If we can't get lettuce from other countries, we produce it ourselves. The independence of it all, i.e., you don't need to depend on somebody for your lettuce and think, or fresh produce like that, and better for the environment. Because obviously, the thing with commercial production or centralized production of crops, pesticides, fungicides, fertilizers, all which obviously have an impact. But the reality is very different. And a few indoor farmers have already found this sort of reality is that the economics aren't there. You know, to grow a lettuce in California versus growing a lettuce in Guelph, the cost basically in Guelph is going to be much higher due to the fact of infrastructure. You'll never beat an open field, would you? So they have to be subsidized, and you need energy. And what people are doing now, they're trying to reduce energy needs. And the important thing is, so you don't get any consumer preference. So if we go to a supermarket, you can't see saying grown indoors, sustainable. Not even all. Well, in the US, they can say organic. They can't say it here because organic means outside, natural sunlight, not LED lights. But the point was, with organic produce, you can charge much more than you can do for conventional. And you can't do that with indoor farm crops. And there is this sort of marketing idea where you say, well, promote the company, saying, oh, yes, this is from this company, which is indoor farm sustainability. But at the end of the day, consumers go by, yeah, quality to agree, but always the cost, isn't it? So the thing was, when we started looking at the food safety of indoor farming, we found it to be very complex. Even the term indoor farming, people say, no, you mean vertical farming, sky farming, urban, hydroponics, bioponics. And there's no agreed on terminology, which is a problem because when you start talking about indoor farming, it's so many different things. This is just a map of all the different derivations that you can have in an indoor farming system, from what growing system to use, peat-based, soilless, hydroponics, aquaponics, to the type of crops and the water sources, the water treatment, irrigation method. I won't go through all these, but if you want to get a background in indoor farming, then I recommend go to the FAO website next month when we're having the launch of our famous food safety guidelines for indoor farms. But the reality is what we found, and what probably people have guessed, is that indoor farming is contained. Basically, it is enclosed. If you want to go into an indoor farm, depending on the sort of establishment, one, they won't let you in the growth rooms. They'll show you a window. Other places I've seen literally tell you to get changed into these sort of paper jumpsuits, so you have to go in, and all this kind of thing. But the reality is, when they bring inputs like seeds, they've got no real screening of pathogens. When they bring the growth substrate in, which is basically composted peat manure, they add a formulation to it. Again, basically they might test it, but what you're going to do about it, you need the growth substrate. So rather than going peat-based, they can go with what we call hydroponics. So things like rockwool and jute. So jute is this popular one because it's made of recycled plastics, and as we'll see, it doesn't mean it's safe. And obviously, nutrient solution, our irrigation water, recycling. And I could tell you the story about, say, New Jersey farmer called Bright Farms. Well, they went bust, but they came back again, so to speak, because got bought here over. Their water supply actually contaminated with salmonella because they were drawing the water from outside pond, and they were putting peracetic acid in, but they didn't validate. They didn't know if this treatment was good or bad to disinfect the water. Obviously, it was bad. And I had these hydroponic ponds, about 20 of them, and I think they was 19 was contaminated with salmonella. And obviously that contaminated the crop. Anyway, I'm digressing. So you bring these things on Trojan horses, which means you need control of them. So we talk about interventions now, the risk management options. And the thing is for many years that they did believe that washing was the best thing. We knew in the early studies we did in '95 that washing wasn't cutting it, because we got commissioned by Marks and Spencer's. So they said, why isn't our microbial loading decreasing? And at this time, we looked at this and we looked at the internalized populations, and sure enough, they were internal. Everybody thought till then inside a plant was sterile. Well, there you go. And obviously endophytes came up. But the reality is then the washing wasn't effective. And vacuum cooling didn't help because obviously that draws bacteria inside the stomata and things like that. Not to mention biofilms. So we did studies again back in the 2000s to show that the chlorination was probably one of the issues, as in they use this oxidation reduction potential, which is a general sensor for oxidative power, and it does feedback. So it feedbacks to a box, and if it needs to increase the ORP, it adds more chlorine, adjusts the pH. But what we did is show them that the ORP meter might be showing really good readings over 800 millivolts, which is a high chlorine, but the actual free chlorine concentration was going all over the place. It was depleted in most cases, and I wonder why it didn't work. So the problem with hyperchlorination is that you get disinfection byproducts, and this is the reason why in Europe, especially Germany, they don't use chlorine in water. They say it's more dangerous, the disinfection products like chlorophenols and things like that, than the actual pathogens, which isn't true, but that's what the philosophy is. And the ORP wasn't cutting it. So even though they were saying, chlorinated the water, it looks good, the reality is that the organic loading in water was so high, it was just depleting everything. And as you might know, if you're in the water business, you have this sort of chlorine demand curve. And what happens is that the chlorine reacts with organic matter until you meet the chlorine demand, after which it goes down. And if you get the break point after that, you get free available chlorine, which is working. But the reality is fresh produce wash tanks were working way before the chlorine demand has been satisfied. So basically what's being sequestered. Now, obviously what happened is the FDA looked at this and said we need a sort of think group, something to look at irrigation, wash tank water, how we can improve it. So the thing was, they had the brightest minds, I guess, in North America, and but the problem with them, the leader of this, and I won't say the name because I'll get into trouble, was basically a canning expert. So his expertise was doing canning, and as you could imagine, canning and washing are two totally different things. And I remember going for many of these meetings, and they're kind of walking in circles. And at the end of the day, they said, right, we can't decontaminate fresh produce with washing. We just have to prevent cross contamination. And the FDA were happy with that. The industry were happy with that. But as you can imagine, what they were basically doing is not solving the problem whatsoever. So there have been alternative approaches. The protective cultures, there's this preparation called EM1. So it's called effective microbes, and it's basically a ferment of cane sugar. It's from Brazil, and they apply it to growth substrate. And the theory goes is that the actual bacteria there, which is a mixed consortia, controls pathogens. Well, it should be done for the Trade Description Act because it's not effective at all. They live things, other sanitizers, peracetic acid doesn't work, electrolyzed water doesn't work. All these other sanitizers, different derivations, they didn't work for the simple reason They were basically water-based and washing didn't cut it. So people have looked at gas plasma, again, very impractical, high voltage. I don't know any commercial systems. UV's out because it's very difficult to obviously sanitize crinkly surfaces. But we came up with a hydroxy radical process. So the hydroxy radical process has its origins way back in the Victorian times. But to give you a bit of background, hydroxy radicals are this very transient species. And they're one of the most oxidative you'll see in nature. And in nature, you'll see them in the clouds. So when the sun uh, beams down on the ozone layer, it generates these hydroxy radicals, and people say it's to do with reducing pollution. And if you're a cloud scientist, a cloud being a literal cloud rather than a IT engineer, they talk about hydrogen peroxide in clouds as a benefit and things like this. And it's used, and it obviously occurs in young stars. So the history of it is the fact that Henry Fenton came up with a reagent based on hydrogen peroxide and ferric, and it was generating hydroxy radicals. And he used it to see if you could oxidize things like carbohydrates. Now the thing was is that the hydroxy radical was only theoretical until 1970 when it was confirmed because so transient. But in the 70s, they took the harness the power of the hydroxy radical to actually treat water. So if you've got water that contains pesticides, hard to things that normal water treatment can't do, they have what's called advanced oxidative process, which basically you put peroxide in solution, pass it through UV, hydroxy radicals are generated, degrading your pesticide. In 83, it was started to be used in carton packaging sterilization. And really, they didn't go out thinking the hydroxy radical. They went out saying, we need to get an alternative to Tetra Pak's 30% hydrogen peroxide. So one of my old profs, he actually came up with this treatment where he had hydrogen peroxide at 1 or 2%, and he'd use UV and it could kill spores off. But if you use too much of a peroxide, it started to neutralize itself. But the thing was, it very effective. And I took this idea from him. Well, I won't say took it, that's the wrong term. I worked with him. He retired very soon after I arrived there to say, why don't we use this for fresh produce? And so we started this research actually in Guelph when I first arrived. And it was basically using the same process, hydrogen peroxide mist, ozone, and UV. And this is a commercial system down there. I don't want to curse, but that's the commercial system they had. So the thing was is that it's a very simple system in a lot of ways. You just got UV light, you introduce your hydrogen peroxide mist, ozone gas through lamps, and it just generates these hydroxy radicals that do the job and then degrade to water and oxygen. Now, I think it can propagate. So since the sort of trials over 20 years ago, they've diversified into a lot of different things. So during COVID, for example, it was N95 mass disinfection. You could do it in 30 seconds. Frozen berries, it's so quick, and because you're not washing them, it comes in frozen, goes out frozen. Low moisture foods, microgreens, which I'll talk about, meat and fish, eggs is a very good one. And even got to do the reusable bag disinfection, and they took it into the field to as an alternative to pesticides and things like crates and that, and things like this. So we started looking at, say, using a hydroxy radical process to look at seed and the disinfection of seedlings. And as I mentioned to you, the seed is this sort of weak point in that people grow them, and when you're growing seed, you're interested in yield, purity. Food safety doesn't come to mind. You've got enough to worry about. And so the thing is, though, is that pathogens survive on seed for a long time, over 2 years. And obviously, when you germinate them, be it a sprout house, you get increased number of pathogens, be it microgreens, again, increased number, even baby leaf. So the thing was, is that once you've enabled a pathogen like E. coli to get established on the plant, it goes on the outside of the plant, but also internally, the endophytic population. And once it's internal, no washing or even hydroxy radical process would get rid of it. So the point was is that we developed this treatment then where we basically got seeds. This is a regular seed. We just treated them in that chamber I mentioned where you got hydrogen peroxide mist and ozone. And we found two things happen. One thing is that it stimulated seed germination. That's a tangible benefit. If you could say to a farmer, I can ensure food safety, they'll look at you blank. But if you said, I can increase germination, plant yield, oh yes, we're definitely interested. And that's what sold it, basically. So not only do you get stimulation in germination, this is just radish seed and Asian blend seed, you get a reduction in pathogens, and that's the key point. So you've got tangible benefits with food safety. Now, the thing about seeds is that they can be difficult to disinfect. One, because you've got to preserve the seed and you've got to access all the crevices. So we developed this activated water process, which was basically where you got a hydrogen peroxide solution, some ferrous iron, and activate it with UV. We call it activated water, a bit like gas plasma activated water. But what we do with the UV system is we activate things. So we activate in terms of starting this chain reaction where you're producing free radicals, which is reducing the oxygen, which then oxidizes the cell structures, and it's this sort of progressive decline, which is propagating. And so we tried that with plants, or just steeping mung beans in this case, with activated water. As you can see, it suppresses the growth of pathogens. In the control, you get up to 10 to the 6, which obviously is very high, which is usually normal. So this method using activated water reduces the pathogen risk. And also, as I mentioned, the tangible benefits which industry look for, it increases this, not only the germination rate, but the yield. That's what sells. It's not the food safety, ironically enough. So that's the seed. What about the hydroponic solution? So when people think about pathogens, they think about, well, I can't grow the hydroponic solution, surely. But the reality is they can, for the simple reason it's a salt solution. An E. coli can grow in salt solution, the same as salmonella. We see different behaviors. So salmonella, which is the orange line here, basically grows and then just sticks around, persists. And it's interesting to note that when we look at indoor farm outbreaks, they commonly implicate salmonella, whereas field-grown outbreaks is E. coli. And I always think this could be one of the reasons, because with E. coli, you get a decline, but you do get an increase subsequent after that. But it's interesting how they behave differently. Now, listeria is totally different. It makes it go to initially grow, but then it just dies off or goes on unculturable. So it just goes to show you, not one rule fits all pathogens, and survival's there. But when we apply the AOP process, this hydrogen peroxide and UV, you can eradicate them, whereas just the UV and peroxide alone, you don't really have much impact. So again, it's this intervention method that's available. And the growth substrate. So with a growth substrate, as I say, jute is one of the most common ones. It actually can support the growth of salmonella. We did some trials where we obviously dampened it because you have to rehydrate it. But then we inoculated salmonella and kept it. The salmonella actually grew on it. So you disinfect it first. It was a very good surface to disinfect for the simple reason it's flat and it readily passes through the uh, hydroxy radical unit. As you can see, trace here as well. And even the crops themselves. So the thing with microgreens is that you can't wash them. If you wash a microgreen, the shelf life goes down to 2 days. But what we found is that if you actually get the tray of crops and we pass them through the generated hydrogen peroxide vapor, the UV, and the ozone. Obviously, in commercial, it'd be like a hydrogen peroxide spray and the UV boom going across the crop. Not only did you reduce pathogens, but you increase the quality of the crop by increasing the chlorophyll. And this is I don't know if you can see the image, but this is a crop with a control, no treat. And this is just a treated just prior to harvest. Much more chlorophyll, much more antioxidant, and it looks healthier to my mind. And what you're doing is stimulating the plant defenses. And we found it in kale. So this is kale microgreens where we treated these post-harvest, but again, you're stimulating plant defenses to enhance its shelf life. And with salad greens as well, they're very distinctive. Normally, without treatment, it just like degrades. With treatment, it stays up there. And again, that's another tangible benefit. And we haven't studied this because we're not really plant scientists, we're more food scientists. But the thing is, when plants get stressed, they induce this defense system. They're reducing the respiration, they're thickening the cell walls, they're accumulating antioxidant and things like that. And so they become much more tolerant to oxidative stress, much more tolerant to would-be spoilage microbes, and enhances the shelf life. Again, by doing so, you got tangible benefits. And they even applied this in the field of spinach. So they did get E. coli reduction, but their kind of purpose was to reduce the need of pesticides so they could actually use it instead of pesticides and also stimulate the plant defenses to get shelf life out of it, which they did. All based on the same technology, though. And I think this is my favorite machine. This is the Back to the Future special. And this is actually growing for vine crops. So, vine crop grapes, isn't it? So, with vines, they're very restrict on pesticides. You know, you're only allowed to use so much. You've got to have withdrawal signs. And so what they did is they built it. When I say they, it's CleanWorks is the commercial branch of it. They made this unit so UV lights, you can clearly see, the hydrogen peroxide, which you can clearly see, and ozone gas, and they would go down the vines to increase the sort of quality of the grapes and reduce mildew. And for that, the award got a sustainability challenge. So this is the CleanWorks team. I'm not part of it. Obviously, I'm an academic, but it's good to see how research gets translated into commercial practice, even though they get all the prizes and we don't. But that's life, isn't it? And maybe not related to well, it is related, is avocado. So over here in North America, we like our avocados, but the trouble is they get soft very quickly. So essentially, the Mission Avocado, which is the largest avocado producer in California, they did a trial, and I wasn't expecting shelf life. I thought they were more interested in listeria, but they did this trial where normally, if you don't have a treatment, they just go soft and ripe and mush. But when they passed them through the hydroxy radical, they had this extended shelf life, which is good when you're transporting things. Again, it's not so much reducing the microbial load, but more to do with the physiology of the avocado. And it works with strawberries as well. You know, we've all been there where we've bought some strawberries, next minute they're mouldy. But with this one, again, it's more to do with physiology rather than these inactivated microbes. And I could have shown you some videos of commercial, like especially the indoor farmers are starting to adopt this technology because they see the value of it. And as I mentioned at the start, I think the reason why they are is because they're new. You know, basically they're coming on the fresh slate. They haven't got these preconceptions that, well, lettuce doesn't harbour E. coli, you know, it's an animal thing, or washing does things. And it's quite interesting. I was actually at a processors a few weeks ago, and I was telling him about the technology. But he had a big background in a major fresh produce processor in the US. And even though I was giving him facts, he was saying, well, no, we like our PAA wash and things like that. And you just say, well, you can't change people's minds, but there is commercial units out there, very quick system and things like that. And this is one of the microgreen grower just down the road here. Yeah, they put it through a very dry process, and that's what they want. They don't want to wet the product, but they want to reduce E. coli and other uh, potential pathogens. And just last week, actually, I was up in Montreal, and they've installed a unit there which can disinfect peas and frozen vegetables. Because what we've had over the last few years is two things. One is that listeria has been associated with frozen vegetables, and people say, where does it come from? And what was interesting is that they've got this listeria. Well, they haven't got. We isolated the listeria monocytogenes from a facility, I won't say which one, which has enhanced persistence under freezing conditions. And when they went to look for it in the freezer, they actually found it in the freezer. It didn't grow, obviously, there, but it accumulated because who washes freezer down? Anyway, to the point is that, yeah, we can. This is a hydrogen peroxide vapor being very atmospheric with ozone gas, and the peas, corns, you name it, go through. And so it enhances that. And the other thing with fresh produce or fresh berries is the norovirus, another hepatitis, and things like that. And we've got a research project looking at inactivating those on berries using the thing. So as final thoughts, the reality is, as I mentioned at the top, fresh produce safety in a lot of ways hasn't moved the dial very much in 30 years. They have preconceptions. Obviously, it's becoming much more significant now. Not only this climate change, which increases the persistence of pathogens and probably the distribution of them, but also we're getting much better at detecting outbreaks. As I mentioned, a lot of outbreaks go unnoticed, and there was at least 3 outbreaks the FDA covered up, and it was only by a journalist going in there, Freedom of Information Act, they detected them. And the FDA come back and said, well, the outbreak was over before we detected the source, so it doesn't really matter. But I think it does matter to change the industry. Now, indoor farming is protected, but it's not totally protected. You need the inputs, and as I mentioned to you, in a lot of ways, people see this as a future, and they're probably true as well. They were certainly good in terms of embracing new technology. Like I say, interventions are the key point. You can't do this by testing. You can't do this by sound bites or LGMA standards. You need that sort of step to disinfect, but you've got to give tangible benefits, be it enhanced seed germination rate or be it shelf life. That sells. Not food safety, again, doesn't sell that much. All right, so with that, I never did any of this work. It was all the troops back in the lab, and obviously the funding was, we only could do it with funding, and we got that through the FAO and OMA for NSERC. I think we've hopefully got time for some questions, but I'll hand it back to Martin.
Thank you so much, Keith. That was absolutely fascinating, and I hadn't really quite realised that just how much fresh produce was an issue, because we don't tend to pick it up so much here in the UK. Although I have heard my friend and colleague in Australia, Tom Riley, talk about outbreaks of C. diff in Australia that are related to onions coming from America, which have been fertilised with pig effluent.
I think in the UK though, don't eat much salad. Maybe that's the reason why.
Well, that's possibly also true.
No,
well, we don't get good enough weather to eat salad.
No, it's interesting you say C. difficile, because we had a project on that, and it does get everywhere, and because it spores, I couldn't believe it was onion.
Yeah, they were giving the piglets cephalosporin antibiotic. And then, of course, that was generating C. diff in the piglets, and then the effluents being used on lawns, but also on onions. So that was quite interesting, because it was a strain they hadn't seen before in Australia, and it turned out to be a North American strain.
We guess around.
Yeah, I mean, the seeds was an interesting one, and I had a little question about that. In that, you talked about how pathogens survive in seeds. Do they survive in, say, I buy my spices like cardamom and other spices, would they survive in those? And are they an issue? And maybe dried pulses that you might buy off the shelf, like beans and things?
It's possible. The good thing about beans and everything is that you tend to soak and cook them. But
do spices as well,
And it's very interesting in seed, because, when we think about all these sprout outbreaks, we have a lot with mung bean sprouts and alfalfa sprouts. And I was talking to a seed producer, because obviously nobody likes to say they've got a problem with the seed production. The actual higher prevalence of pathogens is found in things like sunflower seed and other seed. And you would have thought, why am I doing any outbreaks with sunflower sprouts or things like that? So to answer your question, I think especially these days, you can expect pathogens, because they're so widely disseminated, to get onto things like salmonella. They can persist for a long time. They do die off.
My breakfast currently consists of yogurt with a load of chia and other mixed seeds mixed into it, and certainly they're not cooked before I'm eating them.
Oh yeah, but you're here, so it's not so bad. But
to be honest, I could probably do with losing a few kilos as well. Anyway,
there's better ways of doing it. I can show you with that.
There are.
There was a big, well, it's not related, but outbreaks to do with nuts. Less common, but I remember the big one, which was to do with grains, oats especially. And the thing is that people think, well, it's oats and it's flour, it doesn't matter. But pathogens can get around in most unusual places.
And the more intensively we do things, then it's more likely to be an issue. Well, thank you so much, Keith. That was absolutely fascinating. And can I highlight you to the teleclass education topics coming up in the rest of February. There's a little correction onto this slide that's on the end of today's teleclass. Oh no, it's not. Paul's very cleverly done it. Okay, so it's fixed. So next week we have Tu aeruginosa, or not Tu aeruginosa, how significant are Pseudomonads in waterborne healthcare infections? And that's with Professor Helen Rickard and Elaine Cloutman-Green from the UK, working in Great Ormond Street and probably University College. And then on the 19th, we have the Art of IV Line Care with Professor Claire Rickard from Australia. Claire is a well-known expert in many, many good papers on IV line care, so I'd recommend that one to you. And then on the 26th of February, we have a WHO teleclass, which is an Afro-European teleclass, the global situation of infection prevention control and the case for action and investment in improving it. And that's with Professors Benedetta Allegranzi and her colleague. Can I thank our teleclass education patron sponsors, Diversey, Virox Technologies and Gamma Healthcare, without whom teleclass education would not be possible. My thanks again to Keith. My thanks again to you all for listening. And we look forward to hearing you and speaking to you again on a future teleclass.